Viviparity in fishes, structure of kidney of freshwater and marine fishes, and physiology of excretion. Viviparity in fishes. Viviparity is a mode of reproduction in which embryos develop inside the female's body and receive nourishment from the mother, leading to the birth of live offspring. This adaptation is relatively rare among fishes, but occurs in several groups, including certain species of sharks, rays, and teleost fishes. Types of viviparity in fishes. Viviparity in fishes can be categorized into three main types based on the mode of maternal nutrient supply. Histotrophic viviparity. The embryos consume maternal secretions or unfertilized eggs within the oviduct. Example: some species of rays. Lecithotrophic viviparity. The embryos rely on their yolk sac for nutrition without additional maternal contribution. Example: some elasmobranchs like dogfish sharks. Matrotrophic viviparity. The embryos receive direct nourishment from the mother via placental structures or trophonemata. Example: poeciliid fishes like guppies. Evolutionary significance. Viviparity is thought to have evolved as an adaptive strategy for increasing offspring survival in environments where external fertilization and egg-laying may pose greater risks. It allows for enhanced protection against predators, environmental fluctuations, and oxygen deprivation. Physiological adaptations for viviparity. Viviparous fishes exhibit specialized reproductive structures such as oviductal modifications, the development of specialized uterine tissues to facilitate gas exchange and nutrient transfer. Placentation-like adaptations. Some species develop structures akin to placentas, allowing nutrient exchange between mother and offspring. Hormonal regulation. Prolactin and estrogen play significant roles in regulating pregnancy and parturition. Structure of kidney in freshwater and marine fishes. The kidney is the primary excretory organ in fishes, responsible for osmoregulation and waste excretion. Its structure varies significantly between freshwater and marine fishes due to differences in their osmotic environments. Kidney structure in freshwater fishes. Freshwater fishes live in a hypotonic environment where water tends to enter their bodies via osmosis. Their kidneys are adapted to prevent excessive water intake while conserving essential ions. Key structural features include large glomeruli. These help in filtering a high volume of blood to produce large amounts of dilute urine. Long proximal tubules. Actively reabsorb essential ions like sodium, potassium, and chloride to maintain electrolyte balance. Reduced loop of Henle. Freshwater fishes do not require extensive urine concentration, so the loop is relatively short or absent. Example: carp cyprinus carpio exhibits highly efficient ion reabsorption mechanisms to balance osmotic pressure. Kidney structure in marine fishes. Marine fishes inhabit hypertonic environments where they continuously lose water to their surroundings. Their kidneys are designed to conserve water while excreting excess salts. Key structural adaptations include small or absent glomeruli. This reduces the filtration rate and minimizes water loss. Short proximal tubules. Limited water reabsorption to concentrate urine. Specialized chloride cells in gills. These actively excrete excess salts. Example: marine bony fishes like tuna, Thunnus spp, have reduced renal filtration and rely more on gill excretion for osmoregulation. Physiology of excretion in fishes. Excretion in fishes primarily involves the removal of nitrogenous waste products, maintenance of ionic balance, and osmoregulation. The primary excretory products include ammonia, urea, and uric acid, depending on the fish's habitat and physiological adaptations. Modes of nitrogenous waste excretion. Ammonotylism. Most fishes, especially freshwater species, excrete ammonia directly due to its high solubility in water. Example: goldfish Carassius auratus. Ureotylism. Some fishes convert ammonia into urea, which is less toxic and requires less water for excretion. Example: Elasmobranchs like sharks. Urocotylism. Rare in fishes, but found in some species that inhabit extreme environments where water conservation is crucial. Osmoregulation and excretion. Freshwater fishes continuously gain water and lose ions. They produce copious amounts of dilute urine to expel excess water while actively reabsorbing salts. Marine fishes lose water to their environment and must drink seawater. Their kidneys excrete concentrated urine, and chloride cells in the gills help eliminate excess salts. Endocrine regulation of excretion. Hormones like aldosterone and cortisol play a crucial role in regulating kidney function and osmoregulation. These hormones adjust ion transport mechanisms in response to environmental changes, ensuring homeostasis is maintained. Conclusion. The reproductive and excretory physiology of fishes reflects their remarkable adaptability to different aquatic environments. Viviparity in fishes represents an advanced reproductive strategy ensuring higher offspring survival, while the kidney structure and excretory mechanisms demonstrate evolutionary modifications tailored to osmotic challenges in freshwater and marine habitats.